Okay, let's continue. We are now describing the organelles one by one. And then we say the organelles are considered as small organs within the cell. And then we have those organelles with limiting membrane and those organelles without limiting membrane. Okay, so those organelles without limiting membrane, we'll discuss the ribosomes and the cytoskeleton. They don't have a membrane, but the others, they are membrane-bound structures. So let's look at the endoplasmic reticulum. Now, the endoplasmic reticulum is the network of tubular and microsomal vesicular structures which are interconnected with one another. Okay? They are tubular and microsomal vesicular structures which are connected with one another. They are covered by a limiting membrane which is formed by proteins and by layered lipids. The lumen of the endoplasmic reticulum contains a fluid medium called the endoplasmic matrix. It contains a fluid called the endoplasmic reticulum. They are the diameter of this lumen, they are very microscopic. They range from 400 to about 700 angstrom. Okay. So the endoplasmic reticulum, it forms the link between the nucleus and the cell membrane by connecting the cell membrane with the nuclear membrane. Now, there are two types of endoplasmic reticulum which we are going to discuss, namely the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum and the smooth endoplasmic reticulum. Both types are interconnected, okay? Both types are interconnected, the smooth and the rough. They are interconnected by and continuous with one another. Okay, they are connected and continuous with one another. Now, what's the function of this same smooth endoplasmic, the rough endoplasmic reticulum? It is called rough because it is rough in appearance due to the attachment of granular ribosomes. Okay, that's why it's called rough, due to attachment of granular ribosomes. Now, the function of this is synthesis of proteins. Okay. The other function of this rough endoplasmic reticulum is degradation of worn-out organelles. So I've mentioned two functions, synthesis of proteins and degradation of worn-out organelles. Okay. It's able to synthesize proteins because of their presence of the ribosomes. Okay, these proteins usually are secreted from the cells, and we have proteins such as insulin from the pancreatic beta cells of islets and antibodies from B lymphocytes. Okay, now the next is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, it doesn't have granular endoplasmic or it doesn't have granular structures of ribosomes. It is smooth. Okay. So the function of the smooth endoplasmic reticulum, there are four functions. It is involved in synthesis of non-protein substances. It plays a role in cellular metabolism. It is involved in storage and metabolism of calcium, and it's also involved in catabolism and detoxification. So, this smooth endoplasmic reticulum, we haven't mentioned anything to do with the protein synthesis. What it synthesizes mainly are lipids. Okay. So the smooth endoplasmic reticulum is responsible for synthesis of non-protein substances such as cholesterol and steroids. And we know that cholesterol, steroids, they are very important in synthesis of sex hormones or steroid hormones such as aldosterone, testosterone, estrogen, androgens. They all need the cholesterol. So it is involved in de novo synthesis of cholesterol. So this type of endoplasmic reticulum is abundant in cells that are involved in synthesis of lipids and phospholipids. Okay, what are these cells? We have like the adrenal cortex, the gland, which is found on top of the kidneys. We have the gonads. Okay, so it's abundant in these regions. I also mentioned that the smooth endoplasmic reticulum is important in cellular metabolism because it contains many enzymes which are involved in various metabolic processes of the cell. And also the endoplasmic reticulum stores calcium, and it's indeed the major storage of calcium. In skeletal muscle fiber, it releases calcium, which is necessary to trigger the muscle contraction. Okay, so sometimes it's called the sarcoplasmic reticulum in the muscles because it's involved in storage of calcium. And it's also involved in detoxifying, okay, with substances that are cancer-causing carcinogens. Okay, to detoxify is to make them less toxic, make substances less toxic. So you've seen the importance of this smooth endoplasmic reticulum. Okay. And then we have the Golgi apparatus. The Golgi apparatus, the key thing you need to remember is involved in processing, packaging, labeling, and delivering of proteins and lipid. It is involved in processing. What do we mean by processing? Vesicles containing glycoproteins and lipids are transported into Golgi apparatus. And here, the glycoproteins and lipids are modified and processed. What do we mean when you say packaging? Once the substances are processed, materials are packed in form of secretory granules, secretory vesicles, and lysosomes, which are transported either out of the cell or to another part of the cell. Okay, just like the post office. Okay, it sorts out letters and delivers them to different parts, either within the country or outside the country. So even this Golgi apparatus, that's what it does. And then we also said labeling and delivering of materials. The Golgi apparatus sorts out and the processed and packed materials and labels them. It will label this as a phosphate group depending upon the chemical content for delivery. Okay. Hence, the Golgi apparatus is also called the shipping department of the cell. Some of these mechanisms are so complex. That's why you need to study molecular biology and genetics just to go into detail to understand the function of this. But for this level, we won't go there. We'll just say that the Golgi apparatus, they are involved in processing of materials, packaging of materials, labeling and delivering of materials. Okay. And then we have the lysosomes. Where do we find the lysosomes? There they are. And this is a detailed structure of the lysosome. It's the lysosome, we said, it is made up of the membrane. Okay. Lysosome, not ribosomes. Do not confuse with ribosomes. Ribosomes, we said they don't have a membrane. They are without a limiting membrane. But these lysosomes, they have a membrane. So it's made up of a membrane. And then we can see there's a lipid bilayer there. And then we have inside the lysosomes, we have hydrolytic enzyme mixture. Okay. So the lysosomes are formed by Golgi apparatus. The enzymes synthesized in rough endoplasmic reticulum are processed and packed in form of small vesicles in the Golgi apparatus. Then these vesicles are pinched off from the Golgi apparatus and they become the lysosomes. Among the organelles of the cytoplasm, the lysosomes have the thickest covering membrane. The membrane is formed by bilayered lipid material. It has many small granules which contain hydrolytic enzymes, very powerful hydrolytic enzymes. The types of lysosomes we have the primary lysosomes and the secondary lysosomes. What's the difference between primary lysosomes and secondary lysosomes? The primary lysosomes, these are pinched off from the Golgi apparatus. It is inactive in spite of having hydrolytic enzyme. But the secondary enzymes, or the secondary lysosomes, is the active lysosome. It is formed by the fusion of the primary lysosomes with the phagosome or endosome. I'll explain this later on. Okay, so these are the two types of lysosomes we find in the cell. We have the primary lysosomes, which is inactive, and it is pinched off from the Golgi apparatus. And then we have the secondary lysosome, which is active. It's active because it's a combination, it's a fusion of the primary lysosomes with the phagosome. Now, what's the function of these same lysosomes? Very important. They are called the garbage system of the cell because they are able to degrade many substances in the body or in the cells. Okay, they are called the garbage system. It contains about 50 different hydrolytic enzymes, which are called hydroxylases. Okay. Some of these enzymes we have like the proteases, which hydrolyze the proteins into amino acids. We have the nucleases, which hydrolyze the nucleic acids into mononucleotides. We have the amylases, which hydrolyzes the polysaccharides into glucose. We have the lipases, which hydrolyze the lipids into fatty acids. So it's a very important organelle because it's called the garbage system. Imagine a cell having no ways of getting rid of the garbage. Okay, those are serious abnormalities, and they actually exist, lysosomal disorders. Okay. Now, what's the mechanism of this lysosomal function? There are two mechanisms. There is heterophagy. Okay. There are two mechanisms, heterophagy and autophagy. Heterophagy and autophagy. Heterophagy is digestion of extracellular materials engulfed by the cell via endocytosis. Let me repeat. Heterophagy is the digestion of extracellular materials engulfed by the cell via endocytosis. And autophagy digestion and degradation of intracellular materials such as worn out cytoplasmic organelles. Okay, so these are two mechanisms by which lysosomes are able to perform their function. Okay, so let me mention 4 specific functions of lysosomes. 1, degradation of macromolecules. 2, degradation of worn-out organelles. 3, removal of excess secretory products in the cells. 4, secretory functions. Lysosomes are able to secrete things like perforin and enzymes or granzymes, which destroy both viral infected cells and tumor cells. We'll be able to talk about this later on. Okay. And then the other thing is another organelle we can talk about is peroxisomes. Don't confuse the function of the peroxisome and the lysosomes. The peroxisome have a lipid bilayered membrane that controls what enters and exits the cell. Now, peroxisome have a urate crystal or urate oxidase crystalline core with 32 peroxines. Okay, peroxines are peroxisomal proteins that execute peroxisomal functions inside the organelles. So the peroxisome, they have a urate oxidase crystalline core. Urate oxidase, meaning it's an enzyme. Okay. Now, what are the functions of these peroxisomes? I can mention about 5 functions. They break down excess fatty acids. That's number 1. They detoxify hydrogen peroxides and other metabolic products. They are involved in number 3, acceleration of gluconeogenesis. They are involved in degradation of purines to uric acids. We know what purines are. These are nitrogenous bases such as adenine, guanine. These are nitrogenous bases, very important for the structure of the DNA. So peroxisomes are important to degrade these purines to uric acids. They are also involved in the formation of myelin. Myelin, we know it's a covering found around the neurons. They are also involved in the formation of bile acids. Okay. So let's just know about those functions, peroxisomes. They are different from lysosomes. Lysosomes, we say they are more of, they work on immune kind of functions. Peroxisome, they are more involved in chemical detoxification. Okay. Lysosome, they are more involved in killing pathogens or foreign bacteria and viruses. Okay. The next is centrosomes. Centrosomes will not waste much time. Is the membrane-bound cellular organelle situated almost in the center of cell, and it's closer to the nucleus. In terms of structure, it consists of 2 cylindrical structures called the centrioles, which are made up of proteins. Okay, the centrioles. They are cylindrical in structure and they are made up of proteins. And these centrioles are involved in the movement of chromosomes during cell divisions, such as mitosis and meiosis. So they help to move structures. And then we have the mitochondria. Mitochondria is a membrane-bound cytoplasm organelles concerned with the production of energy. In short, it's known as the powerhouse of the cell. There are three things that it does. It produces energy, that is ATP. 2, it is involved in synthesis of ATP. It's more like a repetition, but I'll explain. And also it's involved in initiation of apoptosis. Apoptosis is programmed cell death, or it's able to cause cell suicide, where a cell kills itself. Now, mitochondria, when we say it produces energy, what we mean is that the energy produced during the oxidation of digested food particles like proteins, carbohydrates, and lipids takes place in the mitochondria because it has oxidative enzymes. Okay? And during this oxidative process, water and carbon dioxide are produced. When we say it's involved in synthesis of ATP, the component of the respiratory chain, the complex 1, complex 3, complex 4, and complex 5 are found within the mitochondria, the ATP synthesis. Okay. Then when we say it's involved in apoptosis, cytochrome C and second mitochondria-derived activator of caspases are involved in apoptosis. I'll explain later on. Okay, because of the presence of the cytochrome C and SMAC. The spelling for SMAC is like this. The mitochondria contains SMAC, which means second mitochondria derived activator of caspases. Okay, but it's also important to know that the mitochondria is involved in storage of calcium. It's another site where calcium is stored in the matrix. However, we should remember that the endoplasmic reticulum is the major site, and it's also involved in detoxification function.